Okay.
It is visible, sir?
Yes, sir. Yes, sir. It's visible. It's changing. Okay. Again, I'm starting with the today's topic, that is optimization. It is also known as quality by design in pharmaceutical sciences. What is exactly this optimization? Uh, as a pharmacist, we should understand to differentiate between the terms good, better, best, and optimized. Anyone who can tell me that what is the literal meaning of good? Any student from this class who can make me understand that what do we understand by good? Is it a relative term or absolute term? Okay. Good is a basically relative term. When you say a something good, it means that it has a few desirable characteristics than the some other thing. Similarly, better is the more advanced version of good. But what is optimized? Optimized is a word which has optimum values, which have desired values of the things. For example, you want to make a tablet and you want the tablet to be disintegrated within 15 minutes. It should give a dissolution of more than 85% in 30 minutes. You want its friability less than 0.5%. These are your standards. And you want its assay more than 95% but less than 100%. These are your desirable things. What happens in general in what we do, we make a tablet based on our experience. And what we do, we in the end of the study, we analyze the batch, whether the batch is passing or it is of standard quality or it is of substandard quality. Next time when the vendor is changed, the vendor which was providing us lactose, when the vendor is changed, the characteristics of lactose are changed, but your shop floor chemist is the same person. He is weighing the same lactose, but the DT has changed with the change of the vendor. And this time the batch is on the verge of rejection. उसकी वैल्यूज वहां तक पहुंच गई कि जहां पे वो क्या हो सकता था, रिजेक्ट हो सकता था. The problem here is the problem that we don't understand the material, we don't have a better understanding of the methods, and so that we are not able to control the outcomes are the desirable characters. Therefore the concept of QBT came into the science. Sir, can we go to the next slide?
Okay.
Okay, I think I am able to change, है ना? Then and optimization is the key for development of drug delivery systems also. Your system should be so robust that some changes in the material, methods, or operator, if these are changed, your system should have cushioning effect, your system should have buffering effect, so that the changes are not going to affect the quality of the product. What happens in a pharmaceutical product development process? Lot of variables. The raw materials, है ना? I want any student to enlist the raw materials for a tablet. One by one you can enlist. What are the various raw materials for the tablets? Saurabh, Saurabh, are you able to hear me?
Saurabh, this is Ram. Saurabh, this is Ram.
सौरभ, टैबलेट्स के किसी एक रॉ मटेरियल का नाम बताइए. Hmm? Any raw material of tablets? Shivendra,
Shivendra Dubey.
Saurabh, you're not audible. You can mute yourself. Shivendra, बेटा टैबलेट में रॉ मटेरियल्स कौन कौन से यूज होते हैं, किसी एक का नाम बताएं आप.
Hello. Just you were telling the excipient used in the tablets. So disintegrant
Good, Shivendra. It's a beautiful answer. At least I'm able to listen something from this class because it gives us a feeling that you are able to understand the thing which I am saying. He is very well correct. He has said one thing, disintegrant. The next lead will be taken by me, but try to give the answer so that we can feel that you are engaged in the class. As Shivendra said, that disintegrants. What are the various raw materials for the tablets? It can be the main ingredient is the drug. We purchase most of the time we purchase the drug from a particular vendor because we, for example, in tablets like metformin, where the dose is 500 milligrams in the normal tablet, and the weight of the tablet is around 730 mg. It means you have a minimum scope of 130 mg of the excipients. The first thing is drug. The drug has many variables. The particle size of drug can be a variable. The level of the impurity can be a variable. The crystalline structure of the drug can be a variable. They can be There can be n number of variables in drug only. The second is the diluent. You have to dilute the API with the help of some excipient. These diluents can be calcium carbonate, these can be dicalcium phosphate, these can be lactose, these can be starch. These are the common diluents which we use in the tablets. Then what the diluents also have these properties: the particle size of the diluent, the source of the diluent. For example, calcium carbonate can be obtained from many places. Oyster shell calcium carbonate can be there. It can be from the limestone. It can be from other source. The size, the source, these are going to affect the your final output of the tablet. Apart from that, as Shivendra said, that disintegrant. There can be many disintegrants. Okay, a few of the disintegrants you are knowing. The two very common disintegrants are SSG and CCS, cross carbonyl sodium, and starch glycolate, sodium starch glycolate. These are the commonly employed disintegrant, but other disintegrants uh, are also used. Apart from disintegrants, some tablets need binders. Soluble starch can be one binder. Other binders can also be used like gelatin. Apart from binders, we use diluents and lubricants. Diluents and lubricants are going to coat the granules if it is by wet granulation method, or they are going to coat the sludges. So many material variables. So many material variables. If anything is changed a bit, the whole process is going to be changed. But pharmacist is a very smart person. He is able to make these tablets like anything. आपको लगता ही नहीं होगा कि इन टैबलेट्स में इतनी आसानी से बन जाता है, बट देयर आर सो मैनी वेरिएबल्स which are needed to be understood properly. These are the variables of the raw materials. Then there are processes. There can be lot many processes. For example, when we make tablets in the lab, that is a different scenario. You mix the ingredients, you go for granulation process, and you dry it, oven dry or air dry. Then you put it in the hopper, mix it with the lubricants, and then you punch it. These are the common steps. But when we go to the industrial scale, you are not going to mix the things in the bag. You are going to use the mixers. Then the problem will be different. In the pilot scale up batch, the mixture is different, and in the production batch the mixture is different. आपको लगेगा oh, it is not the ideal situation, but it's never the ideal situation. These process parameters: how much humidity should be there, how much temperature should be there, what should be the pressure of the punches, these are going to affect the whether the machine is B type tooling, D type tooling, BD tooling, DD tooling. These are going to affect the final quality of the product. Therefore variability. और जब हम इसमें फंसते हैं, मेटफॉर्मिन के साथ सबसे बड़ी प्रॉब्लम है, फॉर मेटफॉर्मिन टैबलेट्स द डिसइंटीग्रेशन इज द इशू. People have reported that they have ingested the metformin tablet and they were able to see the metformin tablet in the स्टूल, वो डिसइंटीग्रेट ही नहीं हुई, पूरा एलिमेंट्री कैनाल पास करके वो बाहर आ गई है, because with the time what happens, it goes on settling. Near to expiry date, it becomes a kind of rock structure which is not going to, you can say, disintegrate. Similarly, some scientists, what they do, their drug is basic, and the disintegrant they are using this, that is acidic in nature. Then the release of drug is very azithromycin, a very common problem. The release is not proper. These are the concerns which need proper understanding. Therefore, variability is the key issue. Anything can be a variable. You can see how many variables: API, active pharmaceutical ingredient variability. The API can be a big variable. The excipient variability. What are the excipients? As Shashank sir already said you that you should understand the excipients. One of the students also told at least one excipient. I am so happy. Disintegrant. So you should understand the variability of the excipient. Process variability. You should understand the process. I have discussed the processes, and in one of the formative assessments, I will ask all these things. What are these processes? Then packaging variability. The primary pack is very important. Your tablet is going to be in touch with the primary pack every time. The variability of the primary pack. I have seen many enteric coating tablets going failed because of the poor choice of the primary pack. And other interactions, there can be some undisclosed interactions, some unknown interactions which are not up to the grasp of a common unskilled mind. Then what happens? The standard deviation square of variance of the product, sigma square is the variance of a product, is basically summative of, it is the integration, it is the addition of, summation of variation of the API, variation of the excipients, variation of the process, variation of the packaging, and variation of the interactions which are available within the various components. What used to happen every time when normal conditions, for example, you are making tablet, what you will do, you will make the composition of drug and polymer. I am using the word polymer here, you can change it to the diluent. Drug and diluent, say for example, in 500 mg of paracetamol, the scope of diluent was only, the scope of total excipient was only metformin, sorry, 130 mg, what I told you earlier. 500 mg की टैबलेट है, 130 mg का हमारे पास स्कोप है, आप उसे 150 कर लीजिए, चलिए अगर पंच के हिसाब से माने हम 7 उसे बना लेंगे 650 की, तो देयर इज स्कोप ऑफ 150 mg. Okay, then in this scope you have to add the diluent. Why we need diluent? Because if we punch the metformin as such, the metformin will settle down and it will form a crystal structure which is not able to break. Therefore, you want to dilute the particles of the metformin with the help of a diluent. The diluent is important. Then what will you do? You have fixed the amount of diluent. Then you will try various amounts of disintegrant. You will take 2% of disintegrant. You will take 2.5% of disintegrant. You will take 3% of disintegrant, and a person can take 5% of disintegrant. But you have changing one variable at a time. In this approach, what we are doing, we are changing one variable at a time. OVAT technique, changing one variable at a time, or OFAT technique, it is also known as one factor at a time, or cost approach, changing one single variable at a time. These are the common approaches which are used in pharmaceutical sciences. We change one variable, we come to its level, then we change the other variable, then we come to the other level. In this way we are going to optimize our composition, but it is not very scientific idea to vary one variable at a time. What will happen when we are working with one variable at a time? There are lot many shortcomings, there are so many problems while changing one variable at a time. What happens? It is a strenuous exercise, it is uneconomical, It involves a lot of money. It is time consuming. It is unsuitable to plug errors when you are okay. अगर ये batch अच्छा निकल गया तो कोई बात ही नहीं है. But what happens after some days, your company changes the tablet operator, or tablet operator gets a good opportunity, he or she shifts to some other company, then what will happen? Everything will change because you don't have a proper understanding of the process and various parameters. It is unsuitable to plug the errors. It is inappropriate to reveal the interactions. It doesn't tell you about the interactions which are available within the various excipients. These are isolated and unconnected studies because you are changing one variable at a time, then you are making it constant. At the next moment, you are changing the other variable, and then you are making it constant. Then you are going to the third variable, then you are making so called optimum. But this optimum is not true optimum. It is a pseudo optimum. Chances अच्छे हैं, if your luck is good then you can reach the true optimum, but there are chances that it is not the true optimum, rather it is a pseudo optimum. Therefore the results which we get are just satisfactory in nature. ये रिजल्ट्स जो हैं बहुत सेटिस्फैक्ट्री, सिर्फ सेटिस्फैक्ट्री, काम चलाने लायक, बिकॉज देयर टैबलेट्स आर एबल टू क्वालिफाई द फार्माकोपियल लिमिट्स. उससे ज्यादा वो कुछ नहीं कर पा रही हैं. Detailed study of all variables is prohibitive. You're not able to understand all the variables in a mechanistic manner, prone to misinterpretation or faking of results because you are not able to understand the things properly. What happens? You are increasing the concentration of super disintegrant up to a level, and after that the disintegration time increases. ऐसे कैसे हो गया? What is a normal understanding of a person that if we increase the concentration of super disintegrant, the dt should decrease, disintegration time should decrease. But after 2.5% in a tablet, you go to 3% and dt is decreasing, sorry, increasing. It is not disintegrating in 15 minutes. Now it is disintegrating in 18 minutes. How it is possible? हमें लगेगा it's not possible, but there are explanations if you are going for a quality by design approach. Futile when all variables change simultaneously. Obviously, you are changing one variable at a time, then it's okay. But some situations come when you have to change all the variables at a single time. For example, the vendor changes, the excipient is changed, but the inspection has given you one notice that the tablets in the market are showing on the average, on the threshold value of dt almost उस वैल्यू को टच कर रहा है, तो आपको फॉर्मूला चेंज करना पड़ेगा. And when you are changing the formula, you are changing all the things at a same time, then all the exercise is futile. Unable to establish cause and effect relationship. क्या किया था और क्या हुआ? This understanding you cannot get from OFAT. But I am not demeaning the OFAT technique, but OFAT becomes good, very good, and best with experience. There are many technocrats who can design a tablet formulation formula in minutes based on their lifetime experience. Therefore, you shouldn't think in mind that OFAT is a totally futile exercise, but that has become the culture that when you are going to praise one approach, then you have to come up with the shortcomings of the other approach. But OFAT is as equal as QBD for an expert person, a person who has a lifetime experience, who has a good understanding of materials, methods, and everything. The OFAT or cost or OVAT technique is equally valid as QBD. In ineffectual as leads to unnecessary runs and batches. Obviously खूब सारे hits and trials करने पड़ते हैं हमें. Therefore when you are stuck up, जब हम फंस गए हैं तब, if it's giving good results, it's okay, but these results are not exactly optimum results. These are pseudo optimum results. Irreproducible. I have told at in many ways that the approaches can be reproducible. The new product may retain defects. You have made a new product based on the previous knowledge. There might be some errors in the or defects in the previous product. These might have retained with when you are moving with the OFAC technique. You please see here. That's very important to see that this drug delivery optimization, what we are doing here, these two graphs are traditional OVAT approaches. You are increasing the gelling powder polymer, and it is the drug release flux. What is flux? Flux is any parameter per unit area. ये सिर्फ हमारा ही नहीं है पैरामीटर, कई फार्मास्यूटिक्स से फ्लक्स हमारा ही है. Any parameter per unit area is known as flux. Okay, so it is the amount of drug released per unit square of the area. So the per unit square of the unit, per meter square, per centimeter square. So you can see as the polymer was increased, initially the flux increased and then the flux decreased. So as the polymer increased, the drug release decreased. The second graph is between the drug release flux and the penetration enhancer. You all know what are penetration enhancers. I can ask someone to give me some names of the penetration enhancers. Anyone who can tell me name of any penetration enhancer? Any student? हाँ, गुड, वेरी गुड. क्या नाम है बेटा आपका? हाँ, नेमतुल्ला. शाबाश बच्चे. So he said that surface surfactants, surface active agents, they are also penetration enhancers. But when you are making, you can understand from this graph that some thing these are not these graphs are not dealing with the tablets. They are more dealing with the some topical product. देखने में तो ऐसा ही लग रहा है कि these are topical products. Non-anionic surfactants are okay. Many non-anionic surfactants like polysorbates, they are well tolerated by the body. But anionic like SDS or SLS or cationic like cetrimide, they have some irritation effects and some other effects on the skin. So, but example is okay, example is acceptable. As the student said that surfactants increase the penetration of a drug across the skin layers. Okay. Other examples are alcohols. Alcohol doesn't means always ethanol. है ना? Alcohols is category, OH group. So alcohols, these can be glycols, these can be glycerines. They are also good penetration enhancers. Even terpenes. Terpenes are also good penetration enhancers. How do they increase the penetration? They disrupt the bilayer structure of the skin. Temporarily, permanent disruption तो नहीं हमें चाहिए. They temporarily disrupt the bilayer structure of the skin layers, and some of the penetration enhancers also dissolve the hyaluronic acid or the cement of the skin. थोड़ा सा सीमेंट भी डिसॉल्व करते हैं तो उससे भी पेनिटेशन होती है. ओके. सम पेनिटेशन एनहांसर्स वर्क ऑन ऑस्मोटिक ग्रेडियंट प्रिंसिपल. तो ये जो पेनिटेशन एनहांसर यहाँ हमने एग्जांपल देखा है, व्हाट इज इट इज डूइंग व्हेन वी आर इंक्रीजिंग द कंसंट्रेशन ऑफ द पेनिटेशन एनहांसर. You can see beautifully that initially the drug release flux has increased, almost become constant, then it is declining, then it is decreasing, then the penetration is decreasing. But what we are doing here, first of all you have come up with a gel polymer से इस बंदे ने ये select किया, the person has selected this concentration of the gelling polymer. Okay, are you able to see the cursor? I think so. This was used as the percentage. Then he or she has moved with the penetration enhancer. Initially she used 1% of the gelling polymer penetration enhancer, and then moved up to here, say it is 5%, it is 1.5%. What they have done first, they have first fixed the gelling polymer as 1.5%. ठीक है, आई कैन आस्क यू ऐसा कौन सा पॉलीमर है जो 1.5% पे जलिंग देता है, बट आई एम नॉट गोइंग टू आस्क. बट यू शुड अंडरस्टैंड that a pharmaceutics person, when we are going for so-called master's degree, we should know the things. These should be in our tips, I think so. And you are also giving answers. I'm quite motivated from your answers. Say we have fixed this concentration at 1.5, say, है ना? And then we have made the gel and added the penetration enhancer 1%, 2%, 3%, 4%, and 5%. Then we have decided the 5%. Now we have come up with a gel which has 1.5% of the gelling agent and 5% of the penetration enhancer. Come to this picture. These two graphs are coinciding here. It is your optimized formulation जो अभी हमने करी है. These are the percentages of the gelling polymer and these are the percentages of the penetration enhancer. 1.5% of the gelling polymer and 5% of the penetration enhancer is giving this optimized product. But in reality, you can see this beautiful spot here. It is the true optimum. Actually, क्या होना था, यहाँ पे 2% gelling polymer होना था और penetration enhancer कितना होना चाहिए था, 1.75%. But फिर भी ठीक है, it is coming in the design space. This area is coming in the desired limits. अभी भी, but what happen, अगर थोड़ी सी गलती होगी, we will deviate something, then we will go outside the desired limits, our outcomes will be totally affected. That is the cost approach and that is the QBD. QBD never misses the true optimum. अभी तक जो अंडरस्टैंडिंग है, अगर हम इसे सही तरीके से यूज करें, बिकॉज गीगो, यू नो ना गीगो, वी आर यूजिंग सॉफ्टवेयर गार्बेज इन गार्बेज आउट. ये तो कंप्यूटर सॉफ्टवेयर आर लाइक स्लेव्स, यू कैन से लाइक दैट, रोबोट्स. बट यू विल डू, दे विल डू लाइक दैट. आप कहोगे पानी लेकर आओ, पानी हिलाएंगे वो, जरूरी नहीं कि ग्लास में लेकर आए. तो you should have to excel in this domain. You should be able to understand the idea of the QBD when you are working with the softwares. नहीं तो मैंने ऐसे भी कई papers देखे हैं, even I am a learner also, where their true optimum is here from QBD, totally out of the design space. अगर आपने डिजाइन ही सिलेक्ट नहीं किया, you haven't defined the constraints properly, then there are chances that you can miss the true optimum. So that is one of the drawbacks of the QBD. The skill is required, training is required. The person should understand the statistical methods. The person should be very versed with the tools of design of experiments. You can see हमें factor 1 कहाँ तक लेकर आया, यहाँ तक. In the traditional approach, we have come from here to, from there to here, and now factor 2 has taken us to here, and it is appearing as the apparent optimum. हमें लग रहा है कि this is the apparent optimum, but our real optimum, our stoka, our target is this. You can miss by this when you work with OFAT, and you can reach the true optimum when you work with systematic design of experiments. It is also known as QBT, quality by design. You can see how much space. आप यहाँ भी चले जाएं अगर गलती होती है, आप यहाँ भी चले जाएं अगर गलती होती है, still we have so much space of deviation. This is the true optimum, this is the real optimum. Then what scientists are saying all over the globe, I have collected all these things for you to understand that systematic optimization using experimental designs is time and cost efficient, assuring high formulation quality. Abu Izza कह रहे हैं in 1996 that it is a time and cost efficient method. Meanwhile, the quality of the formulation is also assured. Then what Newlord et al. Is saying that experimental designs offer an excellent approach to reduce the charge of time and money by limiting experimentation and improving the quality. The learning edge approach of design of experiments has become an integral phenomena globally in drug delivery, especially in the industrial milieu. It is saying by Professor Bhupinder Singh Bhoop, who is a living legend, who has a very good expertise in design of experiments, and we are very well fortunate that we have training under him. What Podzak said in 2006, that dosage form development using trial and error, that is OVET technique or OFET technique, can be characterized as gambling, which never guarantees identification of true optimum. Sure to nahi hai, we can miss the true optimum, and provides a rather second class formulation. He is very harsh in his words, that he is saying that the product which we are saying that is the best is not the best one, rather it is a second class formulation. What Lewis said in 1999, these techniques are no longer the preserve of a few experts and enthusiasts, but are becoming standard tools in management. अब ये common, it's now a common thing. One cannot prevent himself or herself by gaining this experience, because they are becoming common knowledge these days. Before going in the in depth of quality by design, we should understand quality. What is basically quality? Quality is building excellence in a trait. You are bringing excellence in a particular character. And what is excellence? Excellence is a beautiful defined by Oxford Dictionary, is building quality in a trait. It means quality and excellence are in equilibrium. They are interchangeable. Nothing can be achieved without other. None can be achieved without other. Quality is in consonance with excellence, and excellence assures quality, and quality aspires for excellence. Pharmaceutical product quality. When we go to the pharmaceutical product quality word, it's very complicated thing. It is a function of multiple of variables, numerous variables, as I already told you. What is the pharmaceutical quality? Is the function of the drug substance, API. It is the function of the excipients. It is the function of the process. It is the function of the packaging. And if the person is not very well versed, then it is the function of the manpower also. But pharmaceutical industries are very smart, and they are working for more than a decade. ऐसा error नहीं आता. Pharmaceutical scientists are very well trained. You can find some pharmaceutical operators जो सिर्फ tablet punching machine operate करते, they can design very beautiful formulations. पैरासिटामोल और बाकी फॉर्मुलेशंस तो उनके बनाने के लिए बाएं हाथ का खेल है. दे डोंट नीड अ फॉर्मुलेशन केमिस्ट और अ फार्मास्यूटिकल साइंटिस्ट. They are only 10th or plus 2 graduates, pass outs, who operate the tablet punching machines, and they have a lifetime experience, wonderful experience. They can do wonders. But the product quality, because हम सिर्फ पैरासिटामोल टैबलेट तो नहीं बनाएंगे ना पूरी जिंदगी. We have to go into the market, we have to do various jobs. Even many of you are going to manufacture the vaccines. We have to think, we have to understand the thing, and it is a training process. We should understand that pharmaceutical quality is a very complex parameter, which is a role play of the drug substance, excipients, process, and packaging. But you can see that pharmaceutical product quality is a function of multiple variables. What are these multiple variables? They have been written in the form of M. See the beauty of the author. It is from Professor Bhupinder Singh Bhoop, from one of his papers. What are these input variables? Material, machine, method, measurement, manpower, and Mother Nature. Why Mother Nature? Some errors are out of control. As per our present understanding, we are crediting, or we are assigning nature for these errors. Okay, These are the input variables. Y is the output variable. Y is the function of X. And what are these output variables? These are fully understood variables. इनको हम जानते हैं, जैसे टैबलेट के लिए क्या होगा, डीजो, डीटी, फ्राइबिलिटी, यूओसी, यूनिफॉर्मिटी ऑफ कंटेंट, एसए. These are the very well understood variables. Why? These are output variables. Now coming to the 21st century quality initiative that was taken by the FDA, US FDA. This is Pharmaceutical Quality Assessment System, PQAS. Pharmaceutical Quality Assessment System, PQAS, promoting the science and risk-based approach to drug product regulation. PQAS encourages pharma industry to apply QBD principles to systematic development of drugs and drug products. It doesn't mean that हम Indians exaggerate भी कर लेते हैं कभी कभी. It doesn't mean that any application won't be accepted without so-called QBD जो हमारी QBD है, software based QBD. It's not very true. Your cost approach is also a component of QBD. If it has been done with scientific temperament, proper understanding, then also US FDA is going to accept the application for evaluation. But when you have used the DOE, then their acceptance rates are high, because when you have used DOE, it gives better understanding of the process, product, and output variables. So what US FDA has told us that we have to use the components of QBD in the systematic development of drugs and drug products, even in drugs. Then what is design? What is design basically? Systematic approach to attain quality by requisite planning and execution. It is the statement which is given by Professor Bhupender Singh Bhup in 2011 in one of his papers. And in consonance with it, the Swiss Pharma in 2012 said it is the conscious intent to deliver a product or process with a predefined quality to satisfy the customer. It means the quality is culture. Now the quality has become culture. You're not making a product and assessing the quality at the end. You're making a product with full understanding and you're assured of the quality at the end. Obviously those quality parameters have to be checked, but you have proper understanding of the process, you have beautiful understanding of the various parameters, and you are very sure about the quality of the product. QBD is the newest application of PAT. QBD is a blend of prior knowledge and experimental assessment. Obviously, if you don't know, you don't. What is prior knowledge? Which is already known, which is already available in the public domain, which you have gained from your experience, which is gained written in the papers or in various compendias or journals by various scientists based on their experiences. While designing, analyzing, and controlling pharmaceutical manufacturing process using PAT, QBD is essential to be executed through the measurement of critical process parameters. Please understand what is critical process parameters. There are so many process parameters, है ना? You know also that what are drying time, mixing time, the humidity, the temperature, the punching pressure, the rotation speed, all are these process parameters. But out of these various process parameters, what are the critical ones? Critical means out of n number of parameters, which parameters are going to affect the quality of the product. These are known as CPPs. And these CPPs will affect the critical quality attributes. You should understand these two forms, these two abbreviations. CPP, PPs are process parameters. There can be n number of process parameters. But out of these process parameters, which are more influential than others. These are known as CPPs, critical. And out of the quality attributes, for example, a tablet has so many quality attributes, DT, DISO, UOC, friability. इनमें से critical कौन सा है हमारे लिए? So these are CQAs. So therefore, QBT is known is a function of what we know, what we want to know, what we have learned in past, what are the various things which we can control, जो हमारे control में है, what we agree and what we approve, and we expect a state of control. हम कितना control कर सकते हैं? So these are the various things which are going to design your QBT. The concept, we should understand what is quality first. The definition of quality has been given by dictionaries from time immemorial. People understand this word very beautifully, but it is J.M. Juran. He's a quality expert, and he gave this idea on quality by design in the new steps for planning quality into goods and services by J.M. Juran. What he told, he proposed that the quality could be planned in the first place to avoid quality crisis by building quality into the product. जो मैं आपसे पहले कह रहा था, quality should be the culture. Quality should be in the process itself, so that in the end the product will be of quality nature. So you shouldn't forget this name, J.M. Juran. He was the person who inculcated the idea of quality in the quality by design. What is quality by design? What it is all about? You should understand. It is the rational mindset of doing things right to follow the process. It is conglomerate of scientific principles to steer this philosophy. Adoption of these principles to attain quality in the system by design. QBD. Why to implement QBD? Though we have discussed in our previous slides that it enhanced knowledge sharing. QBD makes you to understand the process better. It improves time to market, obviously. It reduces the cost associated with poor quality. If quality is poor, then there will be recalls and rejects. Then it reduces the cost associated with it. What happens when you go to a pharma store and you ask, give me क्रोसिन और give me डोलो, है ना? डोलो तो अब बहुत मशहूर ब्रांड है, but you don't go for generic paracetamol. Being a pharmacist, you know that that is also the same thing. But why we don't go for generic? Because we have a skepticism that the generic drugs are might be of substandard quality. So if these generic drugs are developed with help of QBD, then this skepticism, ये शक है लोगों का about the generic products, it can be counteracted, it can be taken care of. It minimizes the post approval changes. Obviously, if you want to change anything, then also you have the confidence to change the things in a proper manner. QBD is the crux of pharmaceutical development. It is the desired product performance. It is the design of the product. It is the design of the process. It is the performance of the process. Then what happens? The product quality attributes are learned. Then the process parameters are well understood. Then we control the process parameters and we make the desired product specifications. It will result in a better understanding of the product. It will result in the beautiful understanding of the process. And it is a continuous process. It doesn't mean that it stopped here. It is a continuous process. You keep on understanding the product, process, and their outcomes, their specifications, their control, and you keep on learning and you keep on improving. That is the continuous improvement. QBD, what are the salient steps involved? You should understand what are the various steps involved in QBD. First of all, you should define the target product profile. What is target product profile? I am moving with tablet only, है ना? तो क्या है हमारा टारगेट प्रोडक्ट प्रोफाइल कि जो हमारी टैबलेट है उसमें वेट वेरिएशन मिनिमम हो 5%, its friability should be less than 1%, its DT should be less than 15 minutes, its DISO should be more than 85%, it should show a good uniformity of content but within 98 to 100%. These are the QTPP, quality target product profile. You have thought of a beautiful product. That is the thought process. You are making the quality in your mind. ये दिखेगा नहीं आपको अभी. So QTPP is the target. Then what will you do? You will go for the critical quality attributes. For a good product with a good dissolution profile, good bioavailability, good aesthetic appeals, what are the various quality parameters which you are going to observe? For example, you are making digoxin tablet. Digoxin सुना होगा आप लोगों ने, कार्डियोटॉनिक है ना. थोड़ा सा भी एमजीज में चेंज होता है तो पेशेंट की जान को खतरा हो सकता है. हाईली पोटेंट ड्रग. Pharmacist की responsibility है that he should or she should take a good control of the product as well as process so that the quality should be maintained. Okay. Then what happens? How you will go for what are your critical quality attributes? DTDSO तो हो जाएगा इसमें, but UOC, uniformity of content, is very critical. So you have to define your critical quality attributes. What are the there are various quality attributes. All quality attributes are important. But what are the indispensable ones? जिनके बिना move नहीं किया जा सकता आगे. These are CQAs, critical quality attributes. Then what you are doing now, you have to link the material attributes. Material attributes means particle size of the drug, crystalline form of the drug, the type of the disintegrant you are using, the type of diluent you are using, the various properties of the lubricants and glidants. You have to link the material attributes with the process parameters. You have to link the material attributes with the process parameters. You are going for direct direct compression, or you are going for wet granulation or dry granulation. You have to think that what are the various process parameters, and now these process parameters have to be linked with the CQS, critical quality attributes. Then you will have a beautiful design space, the total area where you can work. It's not the exactly area, it is the constraints. From this binder, this percent of binder to this percent of binder, from this percent of lubricant to this percent of lubricant, you have the outer space design. This is the design space. In this design space you have total control. You have total control because you understand the process, you understand the materials. Then you improve your product so that you can get the QTPP. QTPP is the quality target product profile. These are various ICH guidelines which have resulted in the development of QBD concept. These are basically Q8, Q9, Q10, Q11 and process analytical technology. QBD is a vital of synergistic pharma development. Q9 is quality risk assessment. Q8 is pharmaceutical product development. Q10 is quality systems. Q11 is development of drug products. And PAT is process analytical technology. Now we should understand the terms. And what I expect from you that apart from understanding the terms, you should be able to define these terms also. The first thing is the objective, the idea, the your outcome, that is QTPP. the property of interest, or the goal of an optimized experiment. The nature of product, what you want a safe and effective dosage formulation, that is your QTPP. Then variables. What are the variables? These are the constituents or process characteristics of a formulation. They can be quantitative or qualitative. I will come to that part. Independent or dependent, influencing its performance. These are various materials or processes which are going to affect the final formulation. They can be quantitative. What does quantitative means? Whether you will use lactose or you will use calcium carbonate, that is quantitative. Qualitative, sorry, qualitative, these are qualitative. Out of these two, diluent, which diluent you will use, lactose or dicalcium phosphate, it is qualitative. But how much of lactose or how much of dicalcium phosphate or calcium carbonate will be used, that is quantitative. Dependent and independent variables. Independent variables are input variables. You cannot you have control of the binder, but when you have added the binder, that is going to affect the diso. So therefore binder becomes independent variable and the dissolution profile becomes the dependent variable, because dissolution profile depends on the concentration of the various excipients or various components which have been used in the tablets. Defectors. Defectors can be critical material attributes, CMAs. Defectors can be CFAs, critical formulation attributes, or CPPs. They can be critical process parameters. All of these are independent variables which influence the response. Response is quality attributes. For example, amount of binder, that is a material. CMA, critical material attribute in QBD terminology. Some people treat QBD as FDB, formulation by design. कुछ साइंटिस्ट QBD को FDB भी कहते हैं, FBD भी कहते हैं. That is known as formulation by design. In that case, the CFAs is used, critical formulation attributes. Okay, it is a general term for, it can use material, it can use methods. CPPs are critical process parameters. These are independent variables which influence the response. Levels. What are levels? Values or designations assigned to the factor. For example, you are using the binder from the percentage of 1.5 to 3.5%. These are the two levels. You are using the disintegrant from percentage of 2 to 5%. These are the lower limit. Lower limit 2 is -1. We will code it as -1. And 5% we will code 5% as +1. Okay, these are the levels of the various factors. Responses. You have seen the response word here. What are responses? Responses are CQAs, critical quality attributes. These measured dependent outcome, mathematical or graphical, of the experiments. Okay, they can be the DT, they can be the DISO, uniformity of content, friability. These are the quality attributes. But you have to decide, are you going to assess all the quality attributes? Then all are CQAs for you. Or you are going to go for a selected ones, then they can be the CQAs for you. For example, you have, I'm again saying the names. First is weight variation, number one is number two is uniformity of content, dissolution time, disintegration time, assay. Out of these, which one are important for you? You have to think and you have to make your design based on that.